Aldehyde sugars are always aldoses. If there's a ketone, we call them ketoses. Some will form structures in circular rings. Saccharides do some incredible things. Onto a glucose we add a P to it. ATP energy ought to renew it. Quick rearranging creates F6P without requiring input energy.
I know how much you enjoy my musical selection, so there's another sound of glucose for you. Today we're going to be looking at carbohydrates and lipids. Thing to remember about the carbohydrates and lipids, there are three elements that are involved in both: carbon, hydrogen, and oxygen. In the carbohydrate, they're found in a 1 to 2 to 1 ratio. What this means is it'll be C6H12O6 as an example. These sugars or saccharides are the building blocks, so it could be a monosaccharide, disaccharide, polysaccharide. They function primarily for energy, but they can also be structural support, and they are also used for cell recognition. Before we get into the specific examples and how we make polysaccharides from the monosaccharides, I want to take a moment to look at the food sources that you can consume to get these carbohydrates. What you see on the screen are pretty much unprocessed carbohydrate sources, your fruits, your vegetables. At the top of the screen you'll see some breads. Anything that is processed, your flours, with breads, pastas, they're going to be more complex and some say not as good of a source. Whether you're looking at a specific dietary need or not, what we really have to focus on here is how we build these polysaccharides. Remember in the last video clip we looked at the dehydration synthesis process. The slide you have in front of you shows two examples of monosaccharides, glucose and fructose. The monosaccharide is a simple sugar or single sugar monomer, and it's usually produced through photosynthesis. The disaccharide is one water being pulled out, and examples are lactose and maltose for this. And finally, when you get into the polysaccharides are more than 3 monomers, and they have specific examples that I'd like you to know. These examples are chitin, cellulose, and glycogen. The chitin is the crunchiness on your lobster, your insects. It's the shell. It's also in the cell walls of mushrooms, and the last little picture there shows you that it's also used for the dissolving stitches. Cellulose, I remember because it's celery. Cellulose is found in celery, and it is the tough fibrous molecules that are found in a lot of plants. We can't digest it, so you actually use more energy trying to break it down than the value in celery, for instance. The glycogen is how we store our excess energy. And to keep in mind with this, the energy is when you're carb loading. There is a special regimen of exercise and carbohydrate consumption that you do so that you can build up the carbohydrate in your muscle for some sporting event. Now, this slide that I've put in is for the indicator tests. Carbohydrates have 2 indicator tests. For the monosaccharide or simple sugar test, you use Benedict's. You add it a specific amount, and then you're going to heat it. It will turn from a light blue into like this dark rust orange color. The other test is for a more complex indicator or saccharide. It is a starch, which is a polysaccharide, and you put a Lugol's iodine in, and if it doesn't have it, it's a yellow, and if there is starch present, it's a purplish black. It's very, very easy to see, and these two tests are going to be done in lab, so you do need to know what they look like. Keep that in mind, make a note to the star because you will need it for your tests. We're going to take a few minutes now to look at the lipids. And the lipids, unlike the carbohydrates, while they still have the carbon, hydrogen, and oxygen, there is no defined ratio. It's mostly a hydrocarbon, or carbon and hydrogen put together. Functions on this are long-term storage. They're also a major component of the cell membrane, the phospholipid bilayer. We also have it in our waxes and oils that keep our skin moisturized. It's also an important part with cushioning between joints, as well as a lot of plants use it to protect against water loss. Now, before we go into the actual structure of the lipid, the monomers and polymers, I want to go through the functional groups. These functional groups are carboxyl, hydroxyl, amine, and phosphate groups. There's a fantastic chart in your textbook that you need to know. It's going to be on your quiz, test, and midterm. They have specific identifiers, and these structures are how we can tell what compound of life we're dealing with. For instance, when you're looking at the carbohydrate, you have the 1 to 2 to 1 ratio, carbon, hydrogen, and oxygen. In the lipid, you have the carbon, hydrogen, and oxygen, but it's all askew. It's basically hydrocarbons, the carbon with the hydrogen on either end, and a carboxyl group on one side. This carboxyl group is the COOH. Now, if you take that monomer, fatty acid, and attach it to a glyceride backbone, you can have a monoglyceride, diglyceride, or triglyceride. The diglyceride is extremely important to us because that's part of our cell membrane, but most of us are more familiar with the triglycerides. That triglyceride is where you're looking at the steroids and the fats and the oils that we're going to talk about in just a few seconds. Now, I want you to remember that structure at the top right-hand corner. It has a lot of hydrogen packed in tightly. This is important when you're talking about saturated fats. Triglycerides usually come in oils, fats. Oils are liquid at room temperature, fats are solids. So you have your waxes in with that. The saturated fats are all the fats that have hydrogen completely packed in. It's very difficult to digest, and it's considered less desirable. The unsaturated fats have some double bonds, so instead of having all of those hydrogen in, some of those would be replaced when you have the double bond being created. The polyunsaturated have a lot, many double bonds, and this is considered the healthiest type of fat that you could consume. Now, you have to remember, everything has lipids in, even vegetable oils. Vegetable oil means you're getting the oil from the vegetable itself. When you're looking at these, they have HDL, LDL, high density lipid, low density lipid. The H, I remember it as healthy. H is healthy and L is the least desirable. When you're talking about cholesterols, that's what you need to remember. Here's an example of pretty much the wrong kind of lipids, or the unhealthy, full of the saturated fats, or just the saturated fats. Next slide coming up is going to be what the American Heart Society considers healthier fat or lipid solutions. You're looking at avocados, nuts, peanuts, sunflowers, a bunch of different oils that are, again, better choices. They're polyunsaturated. They are easier to digest and considered a better source of the lipid. You need to have that in your diet. It helps you to stay moisturized, and it also is important for your joint health also.